All right, we're going to talk really briefly about meninges and ventricles. You'll see they're very closely related. Well, there we go. Okay, so meninges, it simply means membranes in Latin, and that's exactly what they are. They're just membranes between the actual, skull, between the bones of the skull and the brain. There are these three different layers of membrane. Some of their functions are that they hold the cortical structure in place, right? So think about if you've had a concussion, or maybe you've been following the news, the football players and soccer players that end up with concussions during play. So the meninges are some of the structures that help hold the brain in place during that jarring motion of the head. But because they're kind of thin layers, if the head has moved too much, then they're not able to hold it in place. But from just simple actions like shaking your head or small falls, they're able to protect the brain from damage that way. There's three, like I said, three different meninges. The first is the dura mater, and it's not mater. I didn't make three spelling mistakes. It's actually mater. That stands for, it doesn't stand for. It means mother in Latin. So dura mater just means hard or tough mother. This, like the corpus callosum, gets its name from dissections when, old-timey surgeons were cutting through the dura mater. It was really tough to cut through, so that's how it gets its name. The arachnoid mater just means the spider mother. That refers to the fibrous, you can see it right here actually. So here's the dura mater. You can see how it's a really thick layer. Then you've got the arachnoid mater. And down here, this is actually the subarachnoid space, which is kind of where the arachnoid mater gets its name. It's this really kind of like spiderwebby looking area down here. This is just all these fibers that connect the thicker layer of the arachnoid mater to the pia mater. And so then between there, that's where you can't even see my hands. So it goes from the thick layer of the arachnoid mater through this like spidery layer. Down here is the pia mater. And then like you can see right here, you've got blood vessels that are laying inside that arachnoid, the subarachnoid space. But anyway, that spider web kind of thing is how the arachnoid mater gets its name. And then last, we have pia mater, which just means tender mother. Mother, because it surrounds and protects the brain, is where that comes from. But pia mater is a very light covering. We'll talk about it a little more in just a couple seconds. If you need help remembering these meninges, DAP or DAP, right? So like DAP, dura, arachnoid, pia. All right. The dura mater, there's two layers to the dura mater, although not really. So the top layer, the most distal layer, is the superficial lamella. And that actually is a periosteal layer. So all of our bones have a tissue covering on top of them. All the bones, most of the bones. And the superficial lamella, while being sandwiched right up against the dura mater, is actually just that periosteal layer. So there's really not two layers of the dura mater. One is really a periosteal layer, and then you get to the meningeal lamella, which is the dura mater proper. There's these pretty important parts of the dura mater here. So see this really thick layer of just white, tissue that goes down between the cerebral hemispheres. These are the falx cerebri and cerebelli. You'll see cerebelli's down here. It's a little less pronounced than the falx cerebri, which is up here. But those just keep the halves of the brain from running into each other and touching each other. There's a couple things there. One is, it protects the brain from banging into itself. Another is actually that it it insulates the brain. So the way that neurons talk to each other is an electrochemical process. There's an electrical component and the chemical neurotransmitter component. But the electrical component, if you didn't have this layer, it could be possible for one half of the brain to, send an electrical current down the axon of one neuron, and then for a neuron over here possibly to pick up on that electrical current. You might get, crosstalk between the hemispheres. So this helps insulate. You also get the tentorium cerebelli, which is right here. So the tentorium cerebelli is pretty important because it actually keeps the cerebral hemispheres from crushing the cerebellum. It kind of acts like a shelf that the cerebral hemispheres sit on. Inside the skull, the dura mater is attached to bone, right? So it, we've got the meningeal lamella, and it bumps right up on the superficial lamella, which is a periosteal layer. So in the skull, it's attached directly to bone. In the spinal column, it's not. It becomes a dural sac that surrounds and protects all of the neurons moving down through the spinal column. And that's something that's important to mention here, is that, remember, we talked about the central nervous system as being things that are encased in bone. The meninges flow throughout the entire central nervous system. So they're not just in the brain, they also go down the spinal column. The only difference here is that in the skull, the dura mater is attached to bone, and in the spine, it's within bone, but it's not attached to the bone. It creates its own kind of just floating layer. You may have heard the term subdural hematoma. So dura mater, that's where we get dural, and then sub is below. So you end up with blood between the arachnoid mater and the dura mater. That's a subdural hematoma. That can cause a problem because if you have too much blood flowing into the space between the dura mater and the arachnoid mater, it'll kind of bulge the arachnoid mater down and start pressing on the brain. So having this kind of pressure is not good. One thing is there's not a lot of pain sensors in this area, and it doesn't cause a ton of symptoms depending on where the subdural hematoma is until it does start causing symptoms. But by the time it does start causing symptoms, you don't have a lot of time to get it treated. If you know the actor played Qui-Gon Jinn, why can't I think of his name? I should have looked this up before I started recording this. Liam Neeson. So his wife, they were on vacation and they were skiing. She fell down. She felt totally fine. And then they went back to their chateau or whatever, and she ended up dying before they could get her to the hospital. She had a subdural hematoma. So it's kind of a silent type thing until it's not. I think you have about 3 to 5 minutes from the time that you realize that something is wrong until it's going to be a permanent problem. All right, so then another thing that you can have go wrong with the dura mater is G-force trauma. Now, again, we see this a lot of times with car accidents, like whiplash kind of things. So the G-force trauma kind of whips the head back, and it can actually cause the dura mater to kind of fall through this hole right here. This is your spine hole. And a better term, a more anatomical term than spine hole, is foramen magnum. That just means large borehole. So foramen means a hole that's been bored. Magnum means large. So it's a large hole. It's where your spinal column goes. And you end up, what happens is the dura mater kind of slips down into the foramen magnum, and it pulls some of the cerebellum down with it. And then you get pressure pushing on the cerebellum from that hard portion of skull that the cerebellum has fallen down through. You can end up with difficulty speaking, swallowing, and facial weakness. This might remind you of ataxia from the last lecture. And that's because it's not exactly ataxia. It mimics ataxia because the same portion of the brain, the cerebellum, is being affected. It's just being affected differently. All right. The arachnoid mater, well, we talked about this, gets its name from the fibers that connect. The veins that serve the dura and the pia are sandwiched inside the subarachnoid space. Thought I had a better picture of the subarachnoid space. This one will do. So you can see how this vein is inside the subarachnoid space. Also in the subarachnoid space is cerebrospinal fluid, or CSF. This is a really important fluid that we'll talk about on the very next slide. You also have something called arachnoid villi, and they protrude through the dura, and they allow the CSF to drain back into the bloodstream. So they come up from the arachnoid mater, essentially, right? So you'll have subarachnoid space here, you've got dura mater up here, and then you've got brain. You've got skull, rather. Sorry, brain down here. So you've got subarachnoid space. The arachnoid villi are going to come up through here. So they're going to kind of push up in through the dura mater. And then what happens there is old CSF kind of comes up out of there and then leaks back into the bloodstream. All right, what is CSF? Well, it's cerebrospinal fluid. I told you about that. But what does it do? It helps cushion the brain. So like we talked about with concussions and things like that, you've got this layer of fluid that helps cushion the brain. Another thing it does is that it actually keeps the brain floating inside of CSF so that it doesn't weigh itself down. So it's kind of always floating, like you can think of like, you know, it's in a pool. It's doing like pool floaties. It's got water wings essentially all the time. So it doesn't just kind of like crush itself under the weight. We have the tentorium cerebelli, part of the dura mater, that keeps the cerebral hemispheres from crushing the cerebellum. That's good. But one other thing that keeps the brain from crushing itself is the CSF, just floating in the CSF. Cerebrospinal fluid also carries nutrients to the brain and then takes old things away. Dead cells, waste products from the cells, it carries all that out of the brain. You might have heard about the blood-brain barrier, about how some drugs don't penetrate the blood-brain barrier. Well, the CSF is what we have because of the blood-brain barrier. So CSF can touch the brain. CSF goes directly to the brain. But blood does not. Approximately 500 milliliters of CSF is produced every day by something called the choroid plexus. And I say mostly because we'll talk that the pia mater also produces a little bit of cerebrospinal fluid as well. But the choroid plexus is your big cerebrospinal fluid production area. And this is going to cycle through approximately every 7 hours. So if you look at the CSF that you have in your system now, and you look at the CSF that you have in your system 7 hours from now, it's a completely different amount of fluid, a completely different group of fluid, I guess I should say. If you look at this right here, you see that you've got choroid plexus lining the entire ventricle here. We'll talk more about ventricles in a second, but you've got choroid plexus on the entire proximal portion of the ventricles. That produces CSF and it comes out. What is CSF produced from? You might be wondering. Well, the choroid plexus actually filters blood to create cerebrospinal fluid. So that essentially is what contributes to the blood-brain barrier, is the choroid plexus. Blood comes in, it gets filtered in the choroid plexus, and then from there, cerebrospinal fluid is produced. A lot of impurities that end up in the bloodstream don't end up in cerebrospinal fluid. And then cerebrospinal fluid can interface back in with blood because it is made from blood. So it just drains back into the bloodstream, carrying all of its waste products and everything with it, which then goes to the kidneys and is filtered out. All right. So here again this is what, I have a better slide for this here. So you can see here, this is the lateral ventricles here, the largest ventricle in your body, which is a paired set actually, or the lateral ventricles. You're going to see this one called the third ventricle down here, and you might be wondering, like, well, where's the first and second? Those are the lateral ventricles, but we don't really distinguish between first and second. I think right is first and left is second, but they're just called the lateral ventricles. So the choroid plexus, remember, runs on the proximal portion, the proximal surface of the ventricles right here, and so that's producing CSF. So then it's going to flow through the lateral ventricles and then through the interventricular foramen. Remember, foramen means a hole. Interventricular means, between ventricles. So it goes between the lateral ventricles and the third ventricle, which is here. If I remove that arrow for a second, you can see that the third ventricle is kind of donut-shaped, sort of like donut that's been sat on, because it kind of pouches out this way, but it does have this like hole shape in the middle of it. So the third ventricle is located between the thalamic nuclei in the diencephalon, and there is there's a connection between the two halves, between the two thalamic nuclei, kind of like the corpus callosum for the brain, and that's what runs through this hole right here. So that's why it's not just one condensed ventricle. There's this interthalamic adhesion that goes between the two. So anyway, it flows through the interventricular foramen into the third ventricle, which is right here. Then it goes down the cerebral aqueduct. This is going to tell you where it's heading, right? It's going to go down through the cerebrum and then to the cerebellum. The fourth ventricle is down by the cerebellum. From there, it just kind of flows out into the subarachnoid space, which, remember, the subarachnoid space surrounds the brain, but all three of the meninges are also present in the spinal column as well. So there is cerebrospinal fluid throughout the spinal cord, and it also has a subarachnoid space. So anyway, once it completes its circulation through the spinal column or around the brain, it goes through the arachnoid villi and then spills out into the bloodstream. Oh, and there's some arrows. Okay, here's another look at all that. This is just a front-on look, an anterior view. So the ventricles look a little different from the front view. They don't look like that C shape that they did from the side view, but these are the lateral ventricles. You can see that they're very large ventricles. This over here is just slightly angular views of the same structures. Here's the third ventricle right here. This is the interventricular foramen that the cerebrospinal fluid is going to move through. The cerebral aqueduct is here. And then you've got the fourth ventricle down by the cerebellum. These little guys right here kind of run off into the cerebellum. This is how cerebrospinal fluid gets into the cerebellum. And it'll come back in and then run down through this central canal. And you might have seen on the other one, you've got the foramen of Magundi, and that's where the cerebrospinal fluid flows out into the subarachnoid space. This is an actual view of what the ventricles look like. So these are the lateral ventricles, or a cross section of the lateral ventricles here. This is going to be the cerebral aqueduct flowing right here. You can see how they kind of go deep inside right here. This is, again, the lateral ventricle. Okay, so now the pia mater, which we haven't talked about yet. The pia mater, it's porous to blood vessels, so blood vessels will kind of interface with the pia mater. I know that we talked about blood-brain barrier, but there are little cells that also, they'll take some things from blood, but they don't allow blood to touch anything inside the brain. So the pia mater is porous to blood vessels, but blood doesn't get to the brain. The pia mater really follows the surface of the brain very closely. In fact, if you looked at the brain, you can see it here. You can't even really tell that pia mater is on top of the surface, on top of that gray matter surface. It's just like laminated right to it. And that pia mater is attached to the brain surface by astrocytes. The astrocytes are actually the same structures that will allow some blood flow to get towards the brain, but it'll interface with an astrocyte, and then the astrocyte will carry anything that the CSF doesn't provide the brain to the actual brain itself, to the actual neurons inside the gray matter itself. Spinal pia mater, or mater, sorry, will also kind of follow the neurons very closely. It's a little thicker, though, than it is here on the actual brain. And that makes sense. It's not quite as protected. And then, like I said, the pia mater also produces a little bit of its own cerebrospinal fluid. So to sum up on our meninges, what you've got here is this is the scalp, and then this is there's a little bit of a periosteal layer here. So this is, if you think about the superficial lamella, this is what the superficial lamella is like, only on the inside of the skull. So this is on the outside of the skull. Then this is the skull right here. Then you've got dura mater. Then you've got arachnoid mater, and the subarachnoid space is just below that, between that and the pia mater. And then down here you've got pia mater. And again, you can't even tell that it's on top of the brain. It's so closely laminated right to it. So if you think about the pia mater, it's like laminated. It's like one of those food saver bags that just kind of sucks onto your food. And then the arachnoid and dura mater are more like just a plastic bag. You're going to throw your groceries into it. They're in it, and it just surrounds it. But the pia mater is really like sucked right onto it. All right, and let me see if there's anything else important about ventricles that I missed. No. All right, we'll move on to blood supply next.